Hello everyone, today we'll be doing the virtual challenge meet number 1 for the science UIL test from 2024 to 2025. This video will cover the 20 biology questions in the exam. Before we get started, I'll let you guys preview all 20 questions so you guys can take them on your own, and then we'll be going over the test itself. These are the questions from numbers 1 to number 6. These are the questions from number 7 through 13. And these are the questions from 14 to 18. And these are the last two questions. Now I'll get started with answering all 20 questions of the exam. Number 1. How can vaccines provide protection from disease? So what exactly does a vaccine do to the human body? We know when you inject a vaccine into your bloodstream, vaccines contain deactivated antigens. So I'll just draw them out. So these are, if the circle is like an activated one, it will attack and cause a bad reaction, an inflammatory reaction, for example, in the human body. A deactivated one is just completely damaged. It's just dysfunctional and will not have the same impact towards the body. However, macrophages, neutrophils, and all other immune cells will still attack these dead antigens, and they will transmit this information to memory cells so that the antigens will be encoded into the memory cell, so that when a real antigen appears, it will be able to be killed and exterminated, because the body does have memory on the real antigen. So let's go over each answer choice based on that. Vaccines cause the body to produce toxins that kill pathogens. Our bodies do not produce toxins that kill pathogens. Vaccines don't make us produce toxins. They train our immune system to attack that pathogen in the future. The chemical components of the vaccine destroy new pathogens as they appear in the body. Again, vaccines don't destroy anything. Vaccines don't coat the toxins. They don't physically destroy the viruses themselves. Letter C. Inactive viral parts in the vaccine stimulate the body to produce immunity to that disease. That is what we talked about. The inactive viral parts are those dead antigens that we've covered in the very beginning. And the immunity to the disease is like what we'll get. Once our bodies are trained to fight the disease, once the antigen, we'll be able to fight it back in the future. So C would be the right answer. Let's look at D and E. Vaccines contain macrophages, which hunt for pathogens. Nope, they contain dead, and like I said before, they contain dead virus. And antibiotics and antivirals in the vaccine kill the pathogens. No, that's also not right. So the answer to number 1 is going to be letter C. Number 2. In water, 2 hydrogen atoms is bonded to 1 oxygen atom. What type of bond is found in water molecules? The main thing you want to really focus on is the word "in" here. This is talking about a bond that's in the water molecule itself, not between water molecules. So if I draw a water molecule and another water molecule, what the question's asking are these bonds over here, not the forces between the water molecules. Actually, that would not be right, but not the forces between the water molecules. So we do not want an intermolecular force in our answer. Our answer should not be an intermolecular force, which are the forces between different atoms. In this, in number 2, A, hydrogen bonds. Water does have hydrogen bonds. Hydrogen bonds actually this right here. Hydrogen bond is between hydrogen and the elements nitrogen, oxygen, and fluorine. In this case, water is H2O. This bond is between hydrogen and oxygen. So that's an intermolecular force, but that's not what it's asking. It's not asking between water molecules. And there's also van der Waals or London dispersion forces between water, but again, that's intermolecular force, so we don't want that. We're down to these 3. Is it ionic, polar, or nonpolar? What do these terms even mean? An ionic bond is where, for example, salt, NaCl, is where the structure will be in a crystal lattice structure. So what that means is you will not see it as molecules, but rather as these crystals. Like you'll see in these types of crystals that are branching out. That's how ionic bonds are going to be represented in real life. Water is represented as just H2O. It's not going to be something like this. It's not represented in crystal lattice. So we know it's not ionic based on that. Another way we can tell if it's ionic or not is just the electronegativity difference between oxygen and hydrogen. If I recall, I don't think we will find the electronegativity differences on the periodic table for the UIL science exam. So this one you just got to go based off memory that water is not an ionic compound. So B would be wrong. Between polar and nonpolar is a lot easier to differentiate compared to ionic. Oh, another thing to add is that ionic bonds are formed between metals and nonmetals, and hydrogen is water is formed between 3 nonmetals. So ionic bond, it cannot be B. It cannot be an ionic bond. How we can tell between polar and nonpolar is based on the structure of the atom. So we have hydrogen, we have oxygen, we have hydrogen. Where is, in this bond here, for example, is the electron going to be strictly in the middle? Oh shit, I'm going to draw the electron on the screen to be easier to see. Is it going to be in the middle, or is it going to be closer to hydrogen, or is it going to be closer to oxygen? Which element wants the electron more? This is based on, like I said, electronegativity on the periodic table, and you just have to memorize that oxygen would want it more. Oxygen tugs, so the oxygen will tug on the electron more. You can notate it as this symbol, an arrow with a line. That shows that the electron wants to go to oxygen a little bit more. Same thing happens to this hydrogen. The electron wants to go to the oxygen more. And now you see you have these 2 lines. We want a polar bond is where you can draw a straight line through this, and you can confirm that one side is going to be negative, one side is going to be positive. The electron is on one side, the electron's not. In this case, you can draw a straight line, and you can see that one side is negative, one side is positive. So the answer would be C for number 2. Number 3. A woman with type O blood has a child with a man with a woman with type A blood has a child with a man type O blood. Their child has type O blood. What is the probability that their next child will also have type O blood? So we need to first know what the genotypes of each phenotype or blood type is. If you have the phenotype A, what are the genotypes you'll have? We characterize blood types with the letter capital I and lowercase I. So capital I means that you have either an A or a B. So in this case, we would have a capital I with the letter A, and we have 2 cases. We can either have another capital A, or we can have capital I, lowercase I. Lowercase I in this case, because blood type is both codominant and it's both codominant and normal, like normal dominance. I is the recessive gene. I is the recessive allele, so A will overshadow the I. So these are the 2 genotypes for blood type A. Same thing with blood type B. You could either have IB, or IB with lowercase I. Blood type AB is the codominance. It's where you have both A and B. And blood type O is where you have just 2 lowercase i's. Knowing this, let's go into the Punnett square and let's draw it out. So the woman has type A blood. So we know that the first allele has to be a capital I with a superscript of A, capital A. But what is the second allele? There's 2 cases. We can either have another capital IA, or we can have a lowercase I. So we don't know that. We'll ignore that for now. We know the man is blood type O. So I'll put this as the woman with blood type A, and I'll put this as the man. The man has blood type O. The only possible genotype for blood type O is going to be II. And now it's just simple casework. We know that the first child does have type O blood. If this was capital I, capital A, every phenotype would be this. If you cross them, every one would be capital I, with superscript A and then lowercase I. That would lead to an A genotype, every single child. But we know a child had an O genotype, a phenotype. So this cannot be the case. So that means this is not capital I, capital A. Let's try the other scenario, lowercase I. Cross them together, capital I, capital A, lowercase I. Now you see that there is a 50% chance you can get a blood type O and a 50% chance you can get a blood type A. What is the probability that their next child will also have a blood type O? Is simple 50%. So the answer to number 3 is going to be letter C. Number 4. Which of the following characteristic is a characteristic of the lytic cycle in a virus? So what is the lytic cycle in a virus? So there's 2 different paths a bacteriophage can take when it's infecting a bacteria. The first is the lytic and the second is the lysogenic. So I'll call this lytic, call this lysogenic. The lysogenic cycle is when the bacteria, I'll just draw it as this, injects its DNA into the circular bacteria DNA, and this gets integrated. So I'll just draw it as an integrated DNA. So the bacteria is integrated inside of it. And it stays dormant. Now it just waits until the bacteria multiplies. Copy. It just multiplies. And after several multiplications, then they enter the lytic cycle. The lytic cycle is when a bacteria infects the phage. Remember, it doesn't always have to go from lysogenic to lytic. It sometimes goes straight to lytic. When a bacteria phage infests the cell, maybe not here, but now the cell makes clones of the circular ring. So the phage DNA is going to be, the virus DNA is going to be replicated. And this creates the parts of a virus. This creates the new parts. So these parts start forming the virus again, until the virus once they grow big enough, they just explode out of the cell. So the cell, and then you have all the viruses escaping after cell membrane breaks. And the lysogenic cycle can enter right here. When after some stress, all the cells under the lysogenic cycle will start to undergo the lytic cycle and erupt out of this, and the bacteriophage erupts out of the bacteria. So what is a characteristic of the lytic cycle? A. Viruses remain dormant in the host cell for many generations. That's the lysogenic cycle. The lysogenic cycle is when the virus stays and waits for the cell to multiply. So it can't be A. B. Viral DNA attaches to host DNA and is replicated in successive cell cycles. That is also lysogenic, because successive cell cycles. That's very similar to answer choice A. C. The viral genetic material replicates without destroying the host cell. That is not a property of the lytic cycle, because it will destroy the host cell. Letter D. The number of infected host cells produce a large number of viruses that are released as the host cell is destroyed. That is true for the lytic cycle, because we know the host cell is being destroyed. The lysogenic cycle is when the host cell is dormant for a long period of time, then is destroyed in the lytic cycle. But when you're going strictly in the lytic cycle, that's when the host cell is being destroyed. And E, the infected host cell produces many daughter cells over time, each releasing 2 virus particles per cell cycle. The first part describes only the lysogenic cycle, produces many daughter cells over time, and the second part isn't even true like all the time. Sometimes it can produce more than two. So the answer to number 4 is going to be letter D. Number 5. Sickle cell anemia is caused by a mutation in the gene for hemoglobin. This mutation results in the change of one amino acid from glutamic acid to valine, with all the other amino acids in the protein remaining the same. So make sure to keep that in mind. Based on the info, what type of mutation probably occurred in the gene for hemoglobin? So the main thing you want to keep in mind is all other amino acids remain the same. So if we had a chain, and then we have all the codons. So I'm spacing them out just to like emphasize the triplets. In reality, they're not going to be spaced out in an actual cell. Each codon produces an amino acid. Every 3 nucleotides, remember this is DNA, and each of these line is a nucleotide. Each 3 produces an amino acid. Well, we know that only one amino acid is changed. One glutamic acid is changed to a valine. So what would mean the most likely thing to happen? Translocation of a section of DNA? That's a chromosomal mutation. That's not even, that's the most impactful out of all of these, because that changes chromosomes of that changes whole sections and genes of chromosomes. So that would have changed way more than just one amino acid. Insertion of a base pair. Let's visualize it for a second. If we insert an amino acid, let's just say we inserted this one. That means these 3 are going to be a base pair. These 3 are going to be base pair. These 3 are going to be base pair. And you see how much it changes because of that insertion. Every amino acid is being changed. This one's a new one. This one's a new one. This one's a new one. Everything after the insertion is being changed. So B is not right. What about substitution? When this line is substituted for a new amino acid, how many amino acids are being changed? Just the substituted amino acid, just the substituted codon is the one that's producing a different amino acid. Just one amino acid is being changed, so C would be the answer to number 5. But let's see that D and E. Duplication of a base pair is very similar to the insertion of a base pair. It's essentially insertion of the same base pair. So we can just draw a line. If I duplicate this, remember only these 2 are the same. By the way, even though they're all black, that does not mean they're all the same nucleotides. So I can actually I'll just color code them differently. They're all different nucleotides. If I insert a blue here, what would happen? If I duplicate this one here, what would happen? Instead of having blue, red, black, you'll have blue, blue, red, black, red, green, black, green, green, red, black, red. You're going to have these instead of the normal blue, red, black, red, green, black, and so on and so forth. So duplicating will have the same impact as insertion. And deletion, again, if you straight up removed this blue, like I shall remove that one, you would have complete changing of all the amino acids that follow. So the answer to number 5 is going to be C, substitution. Number 6. When selection acts to eliminate one extreme from an array of phenotypes, it is called what? So let's list out all the types of selections. Natural selection is actually it's not the same as any of these, even though it does have the word selection in it. Natural selection is simply the survival of the species with the best adapted traits. And that is actually not what the problem is talking about at all. Artificial, and we're going to letter E, artificial selection is when humans artificially choose traits to survive, even though those aren't actually beneficial traits. For example, the cuteness of a dog allows cuter dogs to survive longer than ugly dogs. Even though cuteness normally in the environment wouldn't be helpful for a dog, it still determines a factor. between B, C, and D, these are all three types of like selections regarding the phenotypes. Stabilizing selection is when the middlemost phenotype is most present. Disruptive selection is when the extremities of a phenotype are present. And directional selection is when one phenotype is present. One extreme is present. So when you eliminate an extreme, let's just say we had everything was normal, there was nothing, but you eliminated one extreme. So the left side is completely gone. What you instead got was something like this. You got something like this where the left side is very low, the right side is very high. Which of those three graphs would be very similar? I'll actually redraw. This one's really not neat. Which of these three graphs would show that? Where the right side is bigger. That would be directional selection. Directional selection is when one extreme is being favored. So when you're eliminating one extreme from an array, everything is shifting towards the other extreme. So the answer to number 6 would be D, directional selection. Number 7. Excess fluid that diffuses out of the blood plasma is returned to the cardiovascular system by the what? We need to know the functions of each of these body systems. The excretory system, as you can see from the word excrete, its job is to excrete bodily waste. So urine or feces, these are parts of the excretory system, as that's bodily waste. So it's to remove bodily waste. The integumentary system's job is to provide a barrier for your body against like outside particles. So the integumentary system consists of skin, a hair, and nails. And their job is to make sure not only to protect against foreign outsiders, to keep all the organs inside. It's also to regulate warmth and like regulate heat and temperature in the body. So I'll just like mention parts of it. So skin, nails, and hair. And remember, its job is to make sure that it's to protect the body. The lymphatic system has two different functions. One is to fight diseases, like to help the immune system fight diseases, and is also to maintain fluid levels. So fight disease, maintain fluid. So what about digestive and endocrine? Actually, before that, since it maintains fluid, you can go off with the fact that diffusing out excess fluid diffusing out of blood plasma, that's a form of maintaining fluid. So C would be the right answer. Let's look at the other two. The digestive system's job is to allow us to digest our food, to get the molecules out of our food, which is not what's being talked about. And the endocrine system is just about hormone regulation. So our hormones like adrenaline, epinephrine, norepinephrine, all these hormones are being regulated, but that's not what's being talked about in number 7. So the answer to number 7 would be C, the lymphatic system. Number 8. Liver cells differ from skin cells mainly because we know that different bodily cells differ because they express different genes. All cells, you have to keep in mind, all cells have the same genes. Every cell in your body has the same genes. Your brain cells have the same genes as your skin cells. But it's the expression of these genes that makes them different. The brain cell does not express the same genes as your skin cells. So the answer to number 8 would just be the fact that it'd be letter B, because the expression of these cells decide what the function is. They do not have different number and assortment of chromosomes. They have the same number of chromosomes, and they're all assorted perfectly. They do not use a different genomic coding system for translation. We still have cytosine, guanine, adenine, thymine for translation. They do not contain different genes. We all have, every cell has the same genes. And there are no unique ribosomes that are being at play. So the answer number 8 would be letter B. Number 9. All organisms in the kingdoms Protista, Plantae, Fungi, and Animalia are what? We know all these kingdoms, they have one thing in common. They're all part of the animal. They're all eukaryotes. Remember, there are two types, there are three domains. There's bacteria, there's archaea, and there's eukarya. These are the domains. Kingdoms. The eukarya are broken onto these four kingdoms, which are Protista, Plantae, Fungi, and Animalia. So the answer to number 9 will be that they're all eukaryotes. Heterotrophs are animals that can produce their own food. So plants, for example. But animals and protists cannot produce their own food. Fungi and plants can. Animals and protists cannot produce their own food. So it cannot be A. Autotrophs are animals that have to acquire food. Again, this is the opposite. Animals like us and protists, they can acquire food. They eat their food. But fungi and plants, they make their food. So they don't acquire it. So it can't be C. Prokaryotes are they're organisms that do not have any membrane-bound organelles. So they do not have any Golgi bodies. They do not have any endoplasmic reticulum, and they don't have a nucleus. That's their main thing, is that they do not have a nucleus. They have DNA, but that's not bounded in a nucleus. And multicellular protists are not always multicellular. So sometimes protists can consist of just one cell. So the answer to number 9 would just be B. Number 10. Which cell structure is the site of photosynthesis in eukaryotic cells? So photosynthesis, let's list out the function of each of these structures. Ribosome's function is to form proteins, to make protein synthesis. Lysosome's job is to use acids to break down materials. Chloroplast's job is to make sugars using sunlight to make food. So you can stop right here at chloroplast, because photosynthesis is literally making sugar, making food using sunlight and water and carbon dioxide. So the answer to number 10 would be C. But if you want, you can check out the different other two options. Mitochondria is using the sugar to make energy, making energy from sugar. I'll actually put as ATP. And vacuoles are to store water, and waste sometimes. But most of the time, it's gonna be water. So vacuoles' job is to store water. So none of these are anything related to photosynthesis. Only C, chloroplast, is where photosynthesis takes place. So answer to number 10 is going to be C. Number 11. A type of innate learning that can only occur during a brief period after birth or hatching that allows animals to recognize their caregiver for food and protection is called what? So when an animal hatches, it sees its mother and decides to follow its mother around so the mother can protect the animal and provide it food. That's what the question is talking about. And which of the following answer choices best aligns with that definition? Classical conditioning it's a big one. It's when you pair a neutral stimulus with a sensory stimulus and expose them both at the same time to an animal, such that the neutral stimulus also becomes a sensory stimulus. And I'll give you an example for this one. This is if you've taken AP psychology, you'll learn. I think you'll learn this. It's an experiment done by Ivan Pavlov, where he rang a bell. So I'll draw. He rang a bell at the same time he brought food. So just food for a dog. So here's the thing. The food, when a dog sees food, it's gonna salivate. It loves food. It wants to. It's really hungry and it sees it. He's really excited to eat it. The dog doesn't really do much when you ring a bell. It's like you hear noise, it doesn't respond too much. Especially if it's neutral. If it's loud, maybe he might respond. A neutral ringing bell, he's not gonna care much. But since he will respond to the food, I'll draw that in green. He responds to the food, and the bell was ringing at the same time. If this is continuously gonna happen, he's gonna respond to the bell too. So even when the food is taken away and only the bell is ringing, that ringing is gonna cause the dog to salivate. But is that what's being talked about in number 11? No. It's not talking about the pairing of a neutral stimulus with a sensory stimulus. It's talking about a hatchling identifying its mother after birth for food and protection. So A is incorrect. B is imprinting is essentially when animals tend to follow their mother closely for food and protection. That's exactly what the question is. That's the exact definition of number 11. Habituation is when over long periods of time, you start to feel less of the effects of a stimulus. And a good example of habituation would be smoking. Maybe the first few days, people get that rush. Over long periods of time, smoking won't give you the same rush, so you're going to have to do it more often. And this actually is true for any drug. When initially low doses can make you feel good, a long period of time, those low doses will feel normal. And now you need higher doses to feel good. But that's not what the question is talking about. Operant conditioning is a conditioning of rewards and punishment. For example, let's say the mice has 2 buttons. A mice can choose between a red button and a green button. The green button gives the mice a block of cheese, which obviously is a reward. Mice love cheese. I apologize for my drawing. That will do. The red button is going to shock the mice, and mice do not like getting shocked. So which button, if they click them both, let's just say experimentally initially they click them both, the green button got them cheese, the red button got them electric shock, what button would they click most likely in the future? The button that gave them a reward, or the button that punished them? They would most likely click the green button in the future. That's operant conditioning, when we tend to do things that benefit us instead of doing things that punish us. So that's not what the question is talking about, because this question is only talking about hatchlings looking to their parents. Letter E is trial and error learning, which is exactly what it says, trial and error. That's not what's being talked about in the problem, so it's not going to be that. So answer number 11 is going to be B. Number 12. The sodium-potassium pump of a neuron maintains an electrical gradient across the neural membrane by pumping what into the cell and what out of the cell. So the whole structure of action potential, this is what the whole overbranching topic is, action potential. We know that in resting, this is a neural membrane. We're assuming that this is going to be at rest. This is not what's happening during action potential. So let's just say this is the inside of a neuron, and this is the environment. The environment is going to be more positively charged than the neuron. And the reason for this is because of the sodium-potassium pump. The sodium-potassium pump is a pump, so it uses ATP. So when ATP is used, it moves sodium and potassium against their concentration gradients. Potassium is very concentrated inside the neuron, and sodium is very concentrated outside the neuron. Yet, since this is active transport, the sodium potassium pump is going to pump potassium into the neuron and sodium out of the neuron. And at a ratio, if you've heard, this is at a ratio of 3:2. So for every 3 sodium atoms that are being pumped out, 2 potassium atoms are being pumped in. So as you can see, if you're pumping 3 positive ions out and 2 positive ions in, you're overall pumping 1 positive ion out. So because the overall charge, you're pumping an extra positive ion out. That's why the outside of the neuron at rest is more positive than the inside. So the answer to this question, does it pump both into the cell? No. It does not pump both out of the cell. It pumps sodium out of the cell and potassium into the cell. So everything else is just wrong. The answer is going to be letter D. Number 13. Charles Darwin's theory of natural selection does not include the following. So the theory of natural selection states that organisms that have traits that are better suited for their environment will survive long enough to reproduce and pass on those traits. So a good example of that would be in a very dark environment. Let's just say it's very dark. There's 2 types of mice. There's dark mice, and then there's light mice, light-colored mice. Which mice, and there's also predators. There's also like an eagle. Which mice would most likely survive in this dark environment? The mice that's naturally dark enough to camouflage, or the mice that stands out? The darker mice will survive longer than the lighter mice because their coat color is more advantageous. So over time, there will be way more darker mice. Maybe a couple lighter mice. And after very long periods of time, the lighter mice just get their phenotype is just wiped out because it is so much more advantageous to be a darker mouse in this dark environment. So which of the following answer choices aligns with this? A. Organisms within a species differ from each other in the traits that they possess. That's not necessarily false, because that is relatively true though, because within a species they differ from each other. In a species of mice, for example, like in my example, actually, in my example, we had light color mice and dark color mice. They're both mice, but they can differ from each other in the traits that they possess, and that is what leads to natural selection, actually. So A is false. A is true, so it's not the right answer. B, some traits within a species allow for a greater chance of survival. In this case, having a dark coat as a mice is more advantageous than not. So B is true. C, organisms with favorable traits are more likely to survive and produce more offspring than those who do not possess those traits. Black coat is more favorable than lighter coat. Letter D, successful populations produce fewer offspring to ensure that adequate resources are available for all individuals. That's a little... Does that really relate to natural selection? Successful populations produce fewer offspring? Producing offspring is more of like an R selected, K selected species thing. That's not really related to natural selection. And yes, while this may be true, like maybe not successful populations, producing fewer offspring can ensure that adequate resources are available to all. It does not tie into 2 things. It does not mean just because they're successful does not mean they do this. And 2, that does not have anything to do with natural selection. So D is the one that's false. So that is very likely to be right. Let's look at the last option. Organisms compete with each other for survival. That is 100% true. These 2 dark colored mice and white colored mice, they all have the same species. They all have the same food. They're both competing for food, so they do compete for survival. The answer to number 13 is going to be D. Number 14. An individual with Down syndrome has an extra 21st chromosome. This condition is called an... For all these three terms, let's quickly define them. A monosomy is when you have one less chromosome in a pair. One. The thing is, like, the main thing you need to know in a monosomy is that in a normal human karyotype, we have 23 pairs, and each pair has 2 chromosomes. That's 46 chromosomes in total, 23 pairs. One of those pairs is missing a chromosome. So if you have 23 pairs, let's just say 23, say it's female, this is XY. One of them here, let's say this one is missing one. This is a monosomy. A trisomy is the opposite, is when you have one more chromosome. In a pair. So this one, let's say this one would have 3 chromosomes, when normally you're supposed to have 2 chromosomes in a pair. The main thing to know is that both of these are irregular amounts of chromosomes. This is not supposed to happen. These are actually associated with dangerous conditions, like for example Down syndrome especially. These are not what's supposed to happen. And I'm just going to tie this on to letter D because an aneuploidy is just irregular number of chromosomes. We know in a human you're supposed to only have 2 per pair. So irregular numbers of chromosomes per pair. In a human you're only supposed to have 2 per pair. If you have a pair with only 1 chromosome or a pair with only 3 chromosomes, that is an aneuploidy. And there are 2 types of aneuploidy. And examples of aneuploidy are monosomy and trisomy. So that is, we know if it has an extra 21st chromosome, it has 1 more than in the 21st pair. So that would be trisomy. It would have 3. So this would be the 21st pair. That would have 3 chromosomes. So it can't be A. It is D. Here's the thing in this test. You want to pick the most specific answer. D is right because you do have an irregular number of chromosomes. But trisomy is closer to the answer because it's more specific. Versus aneuploidy is just irregular number of chromosomes. We don't know if you're gaining one or losing one. And we don't know like if gaining more or not. A dyskaryosomy is essentially where the cervix gains an abnormal amount of cells. That's actually very different from what's being talked about in number 14. So irregular cell growth in the cervix. And a polyploidy is, I think this is one that you need to make sure not to get confused with. It's when you have more than 2 chromosomes per pair. So this is main thing I want to note for polyploidy is that it's not dangerous. Plants, for example, have 3 chromosomes per pair. We are diploid, meaning we have 2 chromosomes per pair. Haploid stands for for half the normal number of chromosomes per pair. Diploid stands for 2 chromosomes per pair. Polyploid means you're having 3 or more. So polyploid, like for example, a plant would have something like this. It would have 3 chromosomes per pair. So D would not be right because it's only 1 extra chromosome, not an extra chromosome for each pair. So the answer to number 14 is going to be letter B. Number 15. Why is fruit considered an evolutionary advantage of angiosperm? So what is angiosperm? Angiosperm is any type of plant that flowers and produces fruits. So what is the advantage of a fruit? Fruits have 2 main functions. They protect the seed from like any physical conditions, and they aid in the seed's dispersal to the environment. So based on those 2, you know that the answer is not A, B, and D. And I'll go over in depth why. Fruits provide stored energy for the plants that produce them. Fruits do store energy, but not for the plants that produce them. They store energy to nurture the seeds inside. So A would be incorrect. Fruits provide a reservoir of water for the plants during dry conditions. Again, fruits nurture the seed, not the plant. The plant gets its water from its normal roots, and during dry conditions especially, I might talk about it later, it uses two different photosynthetic cycles to to maintain water reservoir while also doing photosynthesis. These are called the CAM cycle and the C4 cycle. These are just two cycles where even during dry conditions, plants can still photosynthesize. D, fruits increase the absorption of light for photosynthesis by absorbing wavelengths of light that leaves do not. This is the furthest from the answer. Fruits have nothing to do with photosynthesis. They do not participate in it. The only thing that participates in photosynthesis are the leaves of the plant. Now, C and E are the closest because fruits do protect and fruits do aid in dispersal. However, what do fruits protect? Do they protect the egg and sperm, also known as the ovule and stigma, the ovary and the pistil and anthers of a plant? Or do they protect the seed of the plant? They do not protect the egg and sperm. They protect the seed. Big thing you have to keep in mind there. E, fruits aid in the seed dispersal by animals, wind, and water. That's what they do. They aid in seed dispersal, not egg and sperm dispersal, seed dispersal. So the answer to number 15 is going to be letter E. Number 16. During what stage of anaerobic respiration is the most ATP produced? So if you memorize the chart on the I have on the right hand side, you will get to know that the oxidative phosphorylation produces 34 ATP, which accounts for the most ATP produced during cellular respiration. So the answer choice is D. However, obviously memorization is it's not too feasible for sometimes. So the best way I would go about doing this is understanding that aerobic respiration means that it has to be in the presence of oxygen. And respiration is the specific cycle of using oxygen to make sugar. Fermentation. Is fermentation aerobic? Fermentation does not require the presence of oxygen to occur. Is glycolysis aerobic? Glycolysis, though it is a part of cellular respiration, it does not require oxygen to occur. In fact, if you guys know this, fermentation actually is a repetition of glycolysis. It produces NAD plus to repeat glycolysis over and over again. What about citric acid cycle? That is a part of cellular respiration. So we can leave it for now and look at the other option choices. Oxidative phosphorylation is also a part of cellular respiration, so we can leave that. And the Calvin cycle is part of photosynthesis, which is not respiration. Throw that out. You're left with option choices C and D. Between these two, you have to probably know that oxidative phosphorylation produces more. The electron transport chain is the bulk of the ATP produced. Citric acid cycle, its main purpose is to produce NADH and FADH too. That's the main purpose of the citric acid cycle. It does produce 2 ATP, but it's not a whole goal. In fact, if you see, per pyruvate, the citric acid cycle produces 3 NADH and 1 FADH2. And it has 2 pyruvates, so that's 6 and 2 respectively. So the citric acid cycle does not account for the most ATP production, so the answer to number 16 is going to be letter D. Number 17. Which of the following is true according to the concept of competitive exclusion? So what is the concept of competitive exclusion? This states that two different species cannot share the exact same niche in an environment, and that eventually one will outcompete and drive the other out of the environment if they're both competing for resources. So that very much aligns with answer choice D, that they cannot share the same niche or role in the environment. For example, if two animals had to hunt bugs, they had to compete against bugs. Eventually, the stronger one will outcompete and drive the weaker species out. Let's look at each answer choice. A. Two species cannot coexist in the same environment. If two species can coexist, that's one thing. But if they're competing with each other, that's another thing. We're not talking about if these two species can compete with each other or not. Like, it's saying two species cannot coexist. What if these two species help each other? What if it's a symbiotic relationship? What if it's mutualism or commensalism, where one actually, like if mutualism, for example, both benefit? That's actually increasing. Like that's not. They're not going to fight each other. So they can both coexist if that's the case. So A is wrong because coexisting is not same as competing. B, the only possible result of competition for resources is the extinction of one or more species. Competition, when there's one species that dominates, competition will drive the weaker species out, but it does not always lead to extinction. That's one thing to mind, is that the only possible result is way too extreme. Maybe a result could be extinction, but most likely the animals will just be driven out of the environment. C, individuals who are best adapted to the environment are better able to survive intra-species competition. Competitive exclusion has to do with interspecies competition. Remember, inter means between. So interspecies competition is between multiple species. Intra stands for within. Intra-species competition is within one species. So within one species, if individuals are better adapted to their environment, that doesn't necessarily correlate to surviving interspecies competition. If all the species are more adapted to their environment, that does not mean they will survive interspecies competition. They'll survive overall. I'd say this statement is relatively true, but two things. Number one, it is not as close to D as being true because D is essentially the definition of competitive exclusion. And C, the main difference is that C is dealing with intraspecies, whereas competitive exclusion deals with interspecies. So C will be wrong for that. And letter E, resource petitioning by competing species will drain all available resources, resulting in the elimination of both species. This is incorrect because one species will dominate and drive out the others, so all the resources will go to the dominating species. So they will not all be drained. So the correct answer to number 17 is going to be D. Number 18. Portions of eukaryotic DNA are transcribed, then removed before mRNA leaves the nucleus to be translated into a protein. These non-coding sections of mRNA are called what? So we want to know the portions of mRNA that have been spliced out because they don't code for anything. After transcription, if you guys remember the mRNA strand, there are both coding parts that are actually useful that will code for the protein, and there are non-coding parts. So the coding parts are called exons. These are the parts that stay during splicing. The non-coding parts are called introns. These are the parts that leave after RNA splicing. So after RNA splicing, you get one segment of spliced and refined mRNA that only contain the exons. The introns are what have been kicked out, so the answer to number A would be introns because those are the ones that are spliced out. The exons are the coding parts of mRNA, so you would not have exons. Inducers actually do not relate to transcription at all. They relate to something called operon regulation. So basically, an operon regulation on a gene. Let's say you have a gene where there's a repressor on this gene. This repressor is preventing this gene from being transcribed. An inducer, I'll draw this a green circle. This inducer, when it binds to this repressor, the complex will leave. So, one sec, whoopsies. It will bind to this, and then this entire complex will leave. So now the DNA is allowed, you can actually transcribe the DNA. But the thing with this, though, is that has nothing to do with RNA transcription as like the non-coding sections of mRNA. This is more related to like before it's been transcribed, how it's been activated, how a gene has been activated. So inducer would not be the answer to this case. Transposons are there are segments in mostly bacterial DNA that move around DNA. So they're not necessarily for transcription, I'd say, in simpler terms. In a circular DNA, if this was the initial region of the DNA, this would move to another region. It would physically translocate into another region. But that has nothing to do with splicing out non-coding sections. As for E, promoter sequence, that is also related to transcription, but that has to do with the sequence of DNA that RNA polymerase binds to. So let's say over here, this sequence of DNA, RNA polymerase binds to it. That is what a promoter sequence is. That first sequence where the RNA polymerase latches to, that is not anything to do with the non-coding sequence of mRNA. So the only answer available to number 18 would be A. Number 19. Which of the following is not true of the carrying capacity of a population? So a population's carrying capacity is the total number of species a population could carry without harming the environment. So if a population exceeds the carrying capacity, that means damage is being done to the environment. So let's look at each answer choices, which is not true. A. Competition with other species can decrease the carrying capacity. If one animal has to fight another animal to get the same resources, and they might end up dying or losing against the other animal, there will be less animals that the environment can carry. So A is true, so it's wrong. B. The carrying capacity of a population is constant over time. There could be so many environmental factors that will throw off the carrying capacity. A volcanic eruption can burn so many plants that now there's less food for animals to eat. That will lower the carrying capacity completely. So no, B is false because there are environmental factors that can change carrying capacity. So B is the right answer. But let's look at the other three. C. The amount of food and water available to a population can change the carrying capacity. This is tied on with what I said in B, where a volcanic eruption destroying food will lower the carrying capacity. So this is true. So C would be incorrect. D. Human activities such as clear-cutting forests, dumping of waste, and suburban expansion can decrease a population's carrying capacity. Yes, if you cut down the trees, some animals can't get their food, or some animals lose their shelters. That can lower carrying capacity because now they have to find it again or risk dying because they don't have it. As for E, an increase in the number of predators in an area can decrease the carrying capacity. More predators meaning higher chance of dying. So less animals can survive. There's a lower chance of animals surviving. And so the carrying capacity of an environment will decrease. So the answer to number 19 is going to be B. Number 20. A scientist observed a dividing cell whose nuclei are reforming and a cell plate is developing along a cell's midline. What stage of the cell cycle is this cell in, and what kind of cell is it in? So we know that there's a cell plate. The cell plate is formed from cellulose and it runs across the cell. So because it's made out of cellulose is only found in plant cells. Cell plates are only found in plants. So A and B cannot be the right answer. As for C, D, and E, we know the nuclei are reforming. If you remember your cell cycle, what phase is it when the nuclei are starting to reform again? We know it disappears in prophase and it reappears in telophase. Additionally, the cell plate of developing across the midline happens in telophase. So the answer to this question would be E. That's it for all 20 questions of the virtual science, the VCM number 1 of the bio section from 2024 to 2025. If you guys have any questions, be sure to leave your questions down in the comments and I will address them as soon as possible. And if you guys like the videos and really think they're helpful, please just put a like down in the video. It shows your support and I would really appreciate it. And I hope I see you guys when I make my chemistry and physics videos for this competition.